Hi, my name is Melody Campbell. I'm an assistant professor here at Fred Hutch in the Division of Basic Sciences. I also have an affiliate position at University of Washington in the Department of Biochemistry. And finally, I'm the scientific director of the Cryo-EM Core here at Fred Hutch. I started my lab about a year and a half ago, so in July of 2020. I came from the University of California, San Francisco. So my lab uses cryo-EM in order to study a family of proteins called integrins. Specifically, we're interested in the integrins that are expressed on the surface of white blood cells, and we're interested in how they work so that we can better understand immunity. Part of the reason why we're so interested in integrins is because it actually has a bunch of different functions. It's implicated in all sorts of functions that have to go on in your body. So if you have issues with your integrins, there can be a lot of really serious downstream effects, like you might get more infections. It's been linked to certain autoimmune diseases such as lupus, as well as certain genetic diseases that make it very difficult to fight off infections. So our hypothesis right now is that the conformation and idiosyncrasies of each individual integrin is really important to its function. And if we know what these unique features are of each integrin, we can presumably design something to change, to alter the integrin's function. However, I always think of structural biology as once you get the structure, that's step one. And then you can interpret the structure and try to figure out what's important, what are the most important parts on that structure. So then when we get the structure, then we can make even more hypotheses, and we are set up in the lab then to express these integrins on the surface of cells and actually test our hypotheses on the cells. So I would say that my path isn't necessarily the most straightforward. So I grew up in a family of musicians. Both my parents played music, and that's exactly why my name is Melody, actually. So in high school, my parents' main focus, for me at least, was to participate in orchestra and spend as much time as possible practicing the violin. So that's always been a part of my life, and I've always been really interested in the arts as well. So when I went to college, I actually started out as a musicology major, and I was interested in going to med school, but over time, especially due to my experiences working in a research lab, I began to realize that that's what I was the most excited about. I started out in a biochemistry lab, but lots of times in biochemistry, the end result would be a series of graphs or a series of gels. But we had a collaboration set up with a structural biologist, and he did cryo-electron microscopy. And that was when I really became interested in cryo-electron microscopy, or cryo-EM, as we often call it. I would hand it off to this collaboration, and usually the next day they would send back either two-dimensional or three-dimensional images of what this protein actually looked like. And that got me really excited, because before it was always just a clear liquid, and you know that the protein should be in there, but you can't actually see anything. So this was really, really exciting that using the electron microscope and a lot of computational techniques as well, we could literally see the protein that I had made. Before the pandemic, I used to do a lot of yoga. That was one of my favorite pastimes when I was back in San Francisco. And then more recently, I've started doing pottery. This I made when I was still in San Francisco, and I'm hoping to be able to start up again now that I'm in Seattle, if things open up again. So what this slide that I'm showing has on it is kind of gives you an idea of what scale you can look at using different ways of visualizing. So just what you can see as a person, you can see everything from fairly large buildings to people, all the way down to about your strands of hair. Then, if you've used a light microscope, you know that if you put samples on that glass slide, you can start seeing things that are even smaller because your images are magnified. You can also start seeing things like pollen, things like bacteria, and even sometimes some of the larger viruses that you can see using a light microscope. Electron microscopes let you look at things that are even smaller than that. So you can use it to look at bacteria, viruses, really small proteins. Some types of electron microscopes are able to visualize individual atoms. So if we want to compare microscopes, This is one of the graduate students in my lab, Adam, and he's using a standard light microscope that maybe you've used before in some of your classes. This is a picture of me with one of the electron microscopes over at the University of Washington. And I always think it's kind of ironic because in order to look at things that are much smaller, you have to use a much, much bigger instrument. Instead of using glass slides, we put our sample on a really small grid. So that's a quarter, and right next to it is the type of copper grid that we use to put our proteins on before we freeze it and put it in the microscope. So the cryo from cryo-EM comes from the fact that these samples are really, really cold, about negative 170 degrees Celsius. So we freeze them in liquid nitrogen. So that's way, way colder probably than anything you've ever felt before. So the sample preparation for cryo-electron microscopy can actually be kind of complicated. So I mentioned before, using biochemistry, we will purify a protein or a complex or a series of proteins. This is kind of an idea of what proteins look like in solution. Proteins are not static, they're constantly moving around. It's one of the things that the lab is specifically interested in. Those proteins, the integrins that are expressed on the surface of white blood cells, they move around a lot. And this is actually really complicated for cryo-EM. So we're trying to work on some methods to help us figure out how to sort out the different conformations of integrins. So what we do is we take a very, very small amount of that sample, 3 microliters, which is actually smaller than a raindrop, and we put it onto that copper grid. Then we load it into an instrument called a Vitrobot, and we hold it between tweezers and very quickly plunge it into liquid ethane with liquid nitrogen surrounding it. So what that does is that it causes the whole sample to freeze so quickly that the water molecules don't have time to form a crystal lattice. And so what you get instead is this glass-like type of ice. And all of your proteins then are suspended inside of that ice. Then what we do, we load this into the microscope, we turn on the electron beam, and it penetrates through the sample. Then what we get is something that we call an electron micrograph, and it looks like this. So you can see that it's black and white, and it's pretty fuzzy. There's a lot of things in the background, but you can also see the individual particles. So something that's really important is that these particles are in different orientations when you freeze them on that grid. Even though you have the same particle all the time, you get these projection images. And so these are just different 2D views of the exact same 3D object. Then we can pick all of the particles, we can align them, and sum them all together. And this is all done using a lot of very sophisticated computer algorithms. We don't actually have to spin these things around and do it by hand. We can basically have the algorithms pick many different orientations of this same 3D object. The algorithms basically sort things out, and then it can project them to make a 3D model from those individual 2D images. If we then zoom in, we can see a lot of structure called secondary structure, which are known to be in proteins. So here we're looking at certain alpha helices within the protein. So this is actually what the state of the art was about 10 years ago. Now more recently, the technology has improved so much that we can almost see individual atoms on the individual proteins. And that's really exciting because it means that for certain small molecule therapeutics or certain drugs, we can make little changes to them to try to help them to bind a protein a little bit better, or really understand why certain genetic diseases come from a single mutation.
My name is Rachel Werther. I am a research technician at Fred Hutchinson Cancer Research Center in the Campbell lab. And my day-to-day work as a research tech is about half of the time sitting at a computer making plans, and about half of the time working with cells and proteins in order to purify the proteins that we are trying to understand. I joined the lab in October of 2020, so it's been about 1.5 years. I actually was not really thinking about science, but instead was just thinking about moving to New York City and just living in New York. And so I made my way across the country. I got very distracted in Colorado working with this project with these civil engineers that were doing a wind power project. I worked on helping build a wind turbine for electrical power in Denver, and then going to India to work with the Indian partners on that project to put up a wind turbine in their village. And then I got really excited about civil engineering. And so I decided I wanted to do civil engineering. That wasn't available in the city I wanted to live, which was Bellingham, Washington. So I decided to do an environmental degree and started going in that direction. And then when I heard about something my friend was learning about in physical chemistry, I thought, that sounds absolutely magic. Why am I not taking that class? And he said, if you took that class, I think you would graduate sooner than with your environmental degree. And I said, sounds good. I joined a lab and I really loved doing bench work. I started looking for work that I could do with structural biology and ended up coming to Seattle. So everything we do in the tissue culture room needs to be sterile. And so we have hoods that make sure that the air isn't flowing in. We wear protective gear so that our skin isn't going to drop living bacteria onto the surface. We spray everything down with 70% ethanol so that none of the living things that can live on surfaces will make their way into our cells. Our cells are growing in a media that's very rich and that almost anything can grow in. So it's very easy for them to get contaminated with bacteria, or any other sort of contamination. So everything we do in here needs to be really sterile, and this is called aseptic technique. This is the flask that we grow our cells in. It's an immortalized cell line, and we store them in these incubators. The incubators are sterile, and the incubators have CO2 and humidity set to a certain amount. And then it's always shaking. Adam is now going to count the cells that he was just looking at. We need to know what density the cells are at because they grow in different phases, and when we want them to grow our protein, we need them to be growing at a certain rate, and that happens when they're at a certain density. We combine the cells with trypan blue, and trypan blue colors dead cells blue and leaves the live cells unstained. And so we mix them, and that's going to let us know the viability of our cells. And then we put it into a cell counter, and that's going to count how many cells we have, so we know what our cell density is. And so what did you find, Adam? What's your density?
That is at 2.51 million cells per ml.
2.5 million cells per milliliter. And so we were hoping it would be a little higher, but that's why we check. Now that he's counted the density of the cells, he can calculate how much of the cell suspension to add to this fresh media in order to get the exact density that he wants. So he's going into the media and being careful not to touch the mouth of the media bottle, and then being careful not to touch the mouth of the flask, so that just in case there's any contamination anywhere, it doesn't get spread. So he's pulling the cells out of the incubator. He knows the exact density of those cells, and he knows how much he needs to add to his pre-warmed media to get the density that he wants. And this is called passaging. So we call this BLI for short, and it's biolayer interferometry. And so we have a light that's sensing the size of the protein on a probe. We're working with these probes, and I'm doing an experiment now that's using 8 of these probes. Each probe goes into a well of this 96 well plate. So the first thing we want to do is make it so that protein A sticks to the probe. We stick the probe into a well that has protein B in it. And if protein B is sticking to protein A, then the way the light passes through, we'll be able to get a signal that tells us that. And so what we see here are the different phases where this is the probe when protein A is starting to stick to it. And so that has been successful. We had different concentrations of protein A in different wells, and where there's a higher concentration, we see a higher signal. I feel like, honestly, if I could have given myself high school advice, it would be exercise more, because I feel like exercise is a pretty good way to cope with stress, and it's really been something that has made a difference in any kind of stress that I've had in my life. Honestly, that is the advice that I would give high school Rachel.
I'm Jeremy Hollis, and I'm a graduate student here in the Campbell lab. I'm in the MCB, Molecular and Cellular Biology. This is my second year, so our first year we do rotations through a few different labs, and then by our second year we've decided on one lab to be in. So this has only been my first year of work in this lab. I grew up in the Washington, D.C. area. I actually moved here from Chicago, though. I moved here for graduate school for MCB. I actually studied evolutionary biology and ecology and theater. I was a double major. So I was out in the field a lot. When I was in school, we had a cool, like little protected arboretum sort of area that we did research in. Yeah, it was a lot of fun. I really like dancing a lot. Probably been doing that for 8 or 10 years, and that's like my favorite way to clear my head. I really like theater, and I'm a set designer for theater, so drawing and painting and architecture. I actually still do that when I have time to pick up a project or two. That's what I was doing when I was in Chicago, and I've started to, now that live performing arts are starting to sort of crop back up, I've picked up a few jobs here and there, which is fun. It really helped learning the molecular side, the small side of evolution. That really changed how I thought about science. I think that molecular approach, the laboratory approach, really helped me say, oh, there's something more here. It's exciting, yeah. But I like that this job is something new and something different every day, because that's, I think, what I've always been hoping for, is to just have something new and exciting to work on every day. And so I think science naturally lends itself to that. Sort of broadly, our lab is interested in the surface of cells and how those cells can make contacts with other cells, and how they decide, oh, this is a good cell, or oh, this is a bad cell, I should do something about it. So we're really interested in how these specific cell surface proteins actually look. And we think that by looking at them, actually really looking at them and understanding what the surface of them is like, we can understand how they might interact with other things. And those small changes that happen on the surface over time, that's what's really exciting to me, because that will tell us, those small surface changes will tell us how that protein might be working, or how it might be different than a similar protein. I'm working on the evolution of this little class of proteins that are on cell surfaces. And those proteins are really interesting because they have evolved in different ways to be able to recognize things that are good, things that are bad, things that are the same thing as the cell itself. In our lab, we actually do a lot of computational work too, so I love being able to switch between like bench work and then looking at something on the computer and figuring out those problems. So we're a structural biology lab, so we're really interested in what really tiny things on cell surfaces actually look like. So that's actually what this is. This is a visual representation of a protein that you would find on a cell surface, which is pretty cool. So sort of stopping right here, this is the actual cell surface protein below, and then this little bit that we see above there is something that it's actually binding to. So this is really cool because we had no idea how these two proteins were talking to each other, and now we have a pretty good start at how it's actually working. And then we can see this density map actually has a lot of detail in it, so we can actually start to see the tiny amino acids, the building blocks that make up the protein. Yeah, so this is a really promising density map, which is great. Great for my project and my and my potential to graduate. What we need to actually get a 3D reconstruction like this is we need that molecule at different orientations on the grid. So we can take pictures of all of those different orientations, sort of average them together a little bit, and then computationally turn them around and do what we call back projecting. And basically that takes the 2D images, puts them all together into a 3D reconstruction. So today I am making electron microscopy grids, and you can think about these as functionally the same thing as a microscope slide. So we take these little grids that I'm pretty tiny here, so much smaller than a microscope slide, of course, but basically what we do is take one of these little grids, put our protein on it, and then today we're going to put a little bit of stain on it, and so that just stains the protein that we're interested in, so we take it to the microscope and can see it. The first thing that we do is something called glow discharging the grids, and this is really pretty to watch, but basically what it does is add a little bit of charge layer to the surface of the grid, and that's what hangs on to the protein, which we need. Otherwise the protein won't stick to the grid. And I need 3 of them today, so I'm just gonna grab 3. Okay. So they're on the slide here. We're just gonna pop that into the glow discharger. Put this back on. So it takes a second to create a vacuum inside. And once it makes that vacuum, it will create plasma inside of this little container, and that will put a thin charge onto our grids. We keep our protein in the freezer at pretty high concentrations. It's pretty dense, but we don't actually want it that dense when we put it on the grids. For the most part, we don't necessarily know actually how dense we want it. So what we do is we make what we call serial dilutions, which is just repeatedly diluting our protein 1 to 10. So in that way, we have a big broad range of different dilutions that we can use, and then we can take all of those to the microscope, figure out what the best protein concentration, we call it, is. So what I'm doing here is just taking those little grids that we had at the glow discharger and using a different set of tweezers, just putting them on like that. We'll just set them up like that. So we'll have a 1 to 10 here, and then a 1 to 100, and then a 1 to 1000. And we can take all of those different dilutions and see what's going to be best. Just did the 1 to 10. Now I'll do the 1 to 100. Like so. What we want to do is get away all of that moisture and then immediately put stain on it so that our protein isn't actually exposed to too much air. So this happens pretty quickly, but take away the water and then stain all in one go. Something that sort of ended up being helpful is that for set design for theater, I do a lot of model making, very fine, like very small building of scale models, and so you have to work pretty delicately with small tools, and those skills ended up sort of transferring pretty well to what I do here for electron microscopy. So you know, if you branch out a little bit and have different interests, sometimes they overlap in fun ways. Really helpful piece of advice someone in my program told me is that even though your experiment didn't work, you did. Like, you went into the lab and you did it, you tried it, and that was really affirming to me. And I think going in to trying something new, like a new experiment or something like that, with that mindset, saying, well, I went in there and now I know what doesn't work and I know what to change, but I still went in there and I did it and I learned something from that experience. That makes those tough days much more bearable, I think.
My name is Kaylee Azumaya. I am the Associate Director of the Electron Microscopy Shared Resource here at Fred Hutch. And on the day-to-day, I just kind of keep this place running. There are 3 of us in the facility. So I manage all of our budgets and administrative stuff, as well as collecting data on the microscopes, training users, and keeping things generally organized. I've been at Fred Hutch since November of 2020, so just over a year. And then I've been in this role for the past 4 months now. Yeah, so new. So I came directly from UCSF, where I was doing my postdoc, so University of California, San Francisco. Before that, I got my PhD at Vanderbilt University in Nashville, Tennessee. And then my undergrad I did at the University of Virginia. So actually, I've been doing electron microscopy since my PhD. So it's been about 9 years now. Whew, okay. Ow. So I've been in the field generally for about 9 years, but while I was in my postdoc, I was kind of trying to decide whether I wanted to be more on the facility side of things, so more supporting the instruments, supporting users who are on them, or whether I wanted to go into a PI position where you run your own lab. So I love to cook, which has similarities to science, right? I actually prefer to cook and not to bake, so I don't really like being exact in the kitchen, which is different than science, or should be at least. I do really enjoy playing volleyball still, though it has been a while and I am wildly out of shape at this point, but I still enjoy it. So in transmission electron microscopy, the electrons actually go all the way through your sample, so you have to get it really thin. So it's either purified proteins, or you have to take a block of tissue and slice it at 70 microns, which is really, really, really small. And then scanning electron microscopy, you can put, a fruit fly is very classic. You can see the fruit fly eye into the scanning electron microscope, and the electrons actually bounce off of it. So you're seeing the surface. So transmission, you can see through things and get that, what's happening in the cells, what's happening in that tissue piece. And then scanning is like what's happening along the surface. I can get really zoomed in and see if there are cracks on fish skin, stuff like that. This is our biggest microscope. It operates at 200 kilovolts, and this one you have to keep your samples at liquid nitrogen temperature the whole time. So that liquid nitrogen tank fills every 8 hours or so automatically. This is what you use for cryo-EM, so this is for protein structure exclusively. So we have the grid in there, it focuses on that grid, and then it goes through and takes 16 images at once. Or in a row, I guess, not at once. And then she can take all of that data and reconstruct it into a protein structure to tell her more about how her protein binds to DNA. We spend about 3 hours doing the setup, and then it's gonna run for hopefully about 12 hours, and then that's the whole data set. And that's why, so Madison doesn't have to sit here for 12 hours. We just need to get it started, so we need that initial 3 hours of setup time, and then she can walk away from it because it's automated. We put the grids into these little holder buttons, so 4 can fit into each of those little guys. And then that's how small they are when they go into the microscope. So the grids have a holy carbon or gold pattern laid over top. Those holes are empty until they have ice frozen in them with proteins in them.
So your grids are inside that little container now?
Yep. So there's a little cassette that holds them, and then this thing just helps the microscope pick them up correctly to go into the microscope straight and everything. So then here, slip it in here. And then it takes it from there. And so what it's doing is it's going down into there and picking that up and then docking it into the microscope itself. And that's what will come in and actually pick up the grids and put them into the column, which sits right here. You can't see anything because they put it in a big box now, but you can open up the one that Jeremy's going to be on.
So this is the smaller of the two microscopes that we use. So we won't do big high resolution data. It's still pretty big, which is funny, but we won't do any of our large data collections for really high resolution stuff on this. This is more just checking to make sure that our sample is good and that we have good conditions and that things look okay to basically move on to the next step. So we're just going to make sure that we have the protein that we're expecting that we have. There's two different computers. This one controls the microscope basically, so all of these hand panels and screens and such will control what goes on here. And then we also have another computer over here that runs a special piece of software that lets us automate all of our data collection. The actual electron, so this is the whole column, and then our sample is right here. The electron source sits at the top of the microscope here. And then there are a few different lenses and a few different objectives that can focus the electrons to that tiny grid where we want them. So this is what the microscope actually looks like, and then the electrons come from up here. I'm just gonna pick up one of the ones that we made. And again, we use these really small pairs of tweezers. So that's that. It's a little hard to see. We put everything against like white sheets of paper basically, so we can see if we've dropped something or anything like that. So this is the holder. This is basically what holds this tiny little grid right here, and we put this into the microscope. But first we have to put our grid in the holder. So I'm just going to put it down for a second and open this up here. So that's put into its holder here, and now we'll take this and we're going to bring it over to the microscope. So the first time I did this, it did not go super great. So we'll just stick it in like that. And it's in. And then a little superstition that electron microscopists have is to tap it a few times for good luck. So we'll do that too. So now that that's in the microscope, everything else we're going to do is on the computer. It used to be that you could actually look at, there used to be a little screen at the bottom of the microscope, so you could actually look at the particles on that screen, but now it's all transferred over here. So what we're actually looking at here is the grid itself. They're a little bit dim, but that circular grid is divided into, is called a grid because it's divided into all of these little squares here. And so what our goal is to find a square that isn't too dark, so something like this or that might be good to look at. And we want to go to that square, and that's going to have our protein on it. So we can move around a little bit to see if we see anything better. And so this is an even more zoomed in view. So our next step is to, it's really important to find the focus or the height of the particles, especially since what we're looking at is really, really tiny. So that's our next step. And to do that, we basically wobble the stage. And so what's actually happening is the grid is going like this. And we want it so that the things that we see on the screen don't move at all. And that tells us that we're basically at the right height that we should be at. So we're going to now use a more automated form of focusing, but this will take a picture and it'll find the focus with the computer, so it'll get much more precise than we ever could, trying to wobble back and forth. We can choose an even tinier spot to focus on. Focusing is everything for this. And then I'm just gonna choose a few areas just to get a sample of the square and see if it'll be good. You can actually start to see some of our little particles just from this focus. Little focus sample right here. So now it's taking exposures right here. And what we're looking for are actually these little tiny specks and flakes all around. That's actually our protein. So like these small things right here. You can see there's a little bit of contamination, these sort of shards. We don't ideally like to see those, but it's okay. We can still process with them because we can computationally pick those out and just focus on the tiny particles that we want. Just looking at it, this is probably the protein that we're hoping for. Like right here, there's a few integrin legs, which we would expect to see. And then our ligand is a little bit more circular, so some of these circular shapes are probably it. The next step would be we would collect maybe 100, 200 of these images, put them all together, and then go through our processing pipeline basically, and that's just picking out all of the individual particles, averaging them together, and then if we start to see shapes that look similar to how we're expecting them to look, then we'll take this sample and we'll move forward to the higher resolution processing. Yeah.
So I think that as a scientist, I've definitely gotten more and more comfortable with things not working. Sometimes things don't work and they're beyond your control, and sometimes things don't work and they're completely because of something you did. I think for me, if I'm really stuck on something in the lab or trying to figure out, or I can't think of good ideas or next steps, the number one thing I do is try to talk to other people. Sometimes being able to talk it out, either I'll figure it out myself or someone will offer some advice that I didn't think of before. If I could give advice to high schoolers, I would say mostly don't worry about it if you don't know what you want to be yet, or if you don't know what you want to do. I remember ever since being really young, people always ask you what you want to do when you grow up, and I never had an answer for that. And I think it's okay if you don't lock into something early. I think it makes sense to try lots of things. Even in undergrad, I was still very, very much figuring out exactly what I wanted to do. So I would say just keep an open mind, and also don't feel like you're behind if you start a little later. And finally, even if you don't get the best grades in something, if that's really what you want to do, you'll figure out a way to make it work.